This video is a continuation of the discussion of the process of translation. In the previous one, we said that the RNA molecules come out of transcription, and we will have three different types of RNA. We have the messenger RNA, which is the long one. We will have the transfer RNA that is very small. It will have an attachment site for particular amino acids. We will have at least 20 different tRNAs because we will use 20 different amino acids. And then the ribosomal RNA that is going to combine with lots of proteins to create the ribosome, which is the machinery where the peptide bonds will be formed. I'm going to play a game, a little bit of the game of peptide, but I will use only the instructional part of this later on, so you guys watch it. It's coming later today, probably tonight. So going back to just review what we have learned during transcription, we remember that we have two different kinds of, let's say, genes. If it is for a prokaryote, we said that it was continuous and we will have no necessity of splicing. But in the eukaryotes, we have introns removed, exons expressed, and that is important to make a synthesis of a protein. And then we also said during transcription that after the RNA is synthesized, the messenger RNA, before it can be used for protein synthesis, first it gets a cap at the beginning by methylation, and then it gets a polyadenine. Now that messenger RNA will have triads, and this triad is what we call codons. It's a triplet, 3 nitrogen bases. Of course, these nitrogen bases are attached to riboses and phosphodiester bond between them, and that will be 4. Let me see how many I have. 1, 2, 3, 4, 5. I have 5 codons, so I have 15 nitrogen bases, 5 amino acids that will be put together making a polypeptide. Now, we also said that there are 3 kinds of RNAs, and I want to explore a little bit of the different parts. First, the tRNA. If we do a little cartoon, that will have the 3' end because it's single strand, and then the 5' phosphate. It is on the 3' end where an amino acid is going to attach. And then we also have the messenger RNA with the triads that we just mentioned a little bit before. And then we will have the ribosome, that the ribosome is made of many copies of rRNAs with proteins to create the machinery where the peptide bonds will be formed. Now, this is another cartoon that I like it a lot because we say that a transfer RNA will have what is called an anticodon. That's an anticodon. So the AGC, this is a codon from mRNA, just by looking at that mRNA, that is AGC. Now, this tRNA is bringing on the 3' end 1 amino acid at a time. Of course, this is already have moved, but let's say this one that has an amino acid. It is CUU. This is the codon in mRNA, and GAA, that will be anticodon, that is bringing 1 amino acid that is going to be attached. Now, the next one, the incoming amino acid. In this case, this is an alanine. The alanine is going to react. The amino end of alanine is going to react with the carbonyl. So you are making an ester with the ACC end of the last tRNA. So that is going to break apart, and that will exit as a free OH, like this one. But before we discuss that with detail, I just want you to understand how we have combination of the tRNAs and the messenger RNA to make the polypeptide. But this is not just those 2 parts, those 2 components. We need more components in order to make the polypeptide bond. So let's go back and look at what is that we say the parts of a transfer RNA. The transfer RNA has the 3' end, that is the site of attachment for the amino acid, and it has the anticodon loop, this is a stem where this is going to combine on this section with the messenger RNA. So if I have a sequence of the mRNA that is going to combine, let me place here AUG, that will have this anticodon will read AUUAGC, using the base pair complementary that we know from Watson and Crick. Now coming back to this transfer RNA, we say that this transfer RNA is of extreme importance because we will have 20 different ones. So there are 20 different tRNAs because we have 20 amino acids, and we know that enzymes are very specific for their substrate. And I want to show you this picture that I got. I like this picture a lot, in which the reaction between the tRNA and the amino acid is the formation of an ester bond. So if you have, in this case, this is the amino acid of histidine that is going to react with the 3' OH of the tRNA. So this is the 3' OH. We're going to make an ester bond right here between this oxygen of the sugar unit and the carbonyl of the amino acid in particular. So the enzyme, this is called a synthetase, and this one is very specific for only histidine, because we know that only histidine is going to have molecular interaction with the enzyme to be put together with the transfer RNA. So that's of extreme importance, the specificity of the transfer RNA. In the following video, because this is becoming too long, it's becoming hard to upload the videos, so they are too long. So in the following video, I will discuss what is that we call the genetic code, in which you will have the combination. You see, this is messenger RNA that has a 5' end and a 3' end, and we are going to combine 3 different nitrogen bases that will codify for one amino acid at a time. The first amino acid is usually AUG, that AUG is the start codon for methionine. If it is in prokaryote, it will be 4-methionine. But that is also the only codon for methionine. In the entire genetic code table that you guys see, there is only methionine one time. So that is start codon, and it's also methionine in any position of the sequence of the polypeptide. It does not mean that all proteins are going to have amino acid number 1 being a methionine, because after translation, after a protein is being synthesized, proteins will be modified. So we will have what is called post-translational modifications.